Okay, let's finish this lecture on plants anatomy and growth by talking about how do plants grow. In particular, what we're going to talk about is the two type of growth that you can observe in plants. The primary growth that all plants undergo and the secondary growth that's only some specific plants, the wood plants, would undergo. Now, why do plants need to grow? Well, plants are very different than animals. They need to grow all the time. They have what we call the indeterminate growth. And why they need to grow is ultimately because they are sessile organisms. They do not move around, right? And so they cannot really wait for the food. They can't go and hunt for food. They cannot go and look for food around. So they have to find strategies ultimately to go and get that food without moving. And the only way they can do that is by keep growing the stems, for example, to get more and more light, to do photosynthesis and produce their own food, or grow their roots so that they can get deeper and deeper and get more water, more nutrients, and so on. So they have this plasticity of growth, ultimately that allows them to grow pretty much in all directions. So in whether it's purely vertically or horizontally and so on. How do they achieve this growth is in two ways. First of all, by increasing the number of cells that they're made of. We'll see when we talk about the meristem how it replicates all these cells, and also by increasing the size of the cells, by making them longer or larger. So that's the two ways in which growth is achieved in plant. Ultimately, now whether we're talking about primary growth or secondary growth, this is all achieved because of the presence of these tissues referred to as meristems. So these are nothing else than groups of undifferentiated cells, basically like stem cells. Imagine like stem cells, like embryonic stem cell type of cells, which can form all the different tissues that are found in plants. All the three tissues that we talked about. Talk about ground tissue, epidermal tissue, vascular tissues. Well, these cells can form all of them. Now, meristems can be divided into primary or secondary. Primary meristems are those responsible for the growth of the main plants. Okay, and they produce what we call the primary growth, first of all, and the primary xylem or primary phloem, the primary tissues in a plant. And this is driven by two types of meristem. We have what we call the apical meristem, and the naming comes from the location. So apical meristems is referred to as apical because it's found at the tip of the shoots or the roots, the stems or the roots, and it drives the growth following the length of the stems and the roots. The second type of primary meristem, which is present only in some monocots, is the intercalary meristem. So not all plants have it. And in this case, it's not found on the tips, but it's found in between nodes. Nodes are those areas where you have the branching taking place. So in the internode space, the space in between, and they cause the growth of the space between the nodes, so the internode. And then there are secondary meristems. These are found only in some plants, called wood plants. They drive secondary growth, and they are established ultimately from tissue left behind from the apical meristem. And what they do, instead of causing the growth of the plants in following the length of the plants, what they do, they cause the growth of the width of the plants. So there are two types of growths. I will somehow introduce them, and they are related to the type of meristems that they are driven by. So there is primary growth. All plants would undergo primary growth. That's what established ultimately the structure of the basic plants. Okay, and it keeps going throughout life. So the primary growth is that growth that follows ultimately the axis of either the stem or the roots. It happens in both structures there. All plants would have it, and it's usually driven by the apical meristem, in monocots also by the intercalary meristem. And then the secondary growth is present only in wood plants, is driven by a second type of meristem, which is the secondary meristem. It's the vascular and cork meristems. We'll talk about that, or cambia. And what it does, instead, it drives the growth of the plants in terms of width of the stems or width of the roots as well. Let's focus first on the primary growth. Again, all plants would have primary growth, and this is driven by the apical meristem. Okay, that meristem, I'll repeat it over and over, that is found at the tip of the stems and of the roots. And what it does, it causes the growth in terms of following the axis of the stems or the roots, not lateral growth. Now, some plants, again, they have intercalary meristem, so which is present in between the nodes. And what it does, basically, this intercalary meristem, by growing and elongating the cells produced, will cause the growing in distance between the nodes, so the growth of the internodes here. Now, how does it work? Well, we have this apical meristem, which is found at the tip. So let's look first at the stems. So you have that at the tip of each stem, you would have this apical meristem, this undifferentiated tissue, which can give origin to all the tissues. So what happens is that now you have that this apical meristem keeps dividing, keeps producing more and more of these undifferentiated cells. By doing that, it will be causing the growth of the stem in a certain direction, following the axis again of the stem. Now, the tissue that is left behind, so it is moving this way, the tissue that's left behind, it would then first differentiate it into 3 meristems. These are still undifferentiated cells, which, however, they are already going towards creating a certain type of tissue. You have protoderm, procambium, and ground meristem that are left behind. So if this was, for example, the apical meristem here, it's moved this way, it's growing this way. So here you start having now these 3 primary meristems: protoderm, procambium, and ground meristem. So here is how they are located. Protoderm on the outside, procambium in between the ground meristem and the protoderm here. And then with the growth going, these 3 primary meristem would differentiate finally in the final tissues, those tissues that we talked about in the first part. So the protoderm, moving on here, the protoderm here would differentiate into the epidermis. The procambium would differentiate into the vascular tissue, so the primary xylem and phloem. This is referred to as primary growth, so these tissues are referred to as primary tissue, so primary xylem and primary phloem. And then the ground meristem here will form the ground tissue, which will cause ultimately the formation of the pit here. These will be parenchymal cells, then you would have sclerenchymal cell formed here as well. So it will form ground tissues here. And so by doing that, basically the plants has been growing in this direction, and the tissue left behind has differentiated into those tissues and structures which are necessary for the plant to grow and survive. Now, the apical meristem often leaves behind some of the tissue, fully undifferentiated meristematic cells, which put together they're referred to as axillary meristem here. These are those basically apical meristems, so axillary meristem just because they are left behind and they form ultimately the branching there, which then can indeed cause the formation of the side stem, so the branches there. Now they can produce both vegetative or reproductive stems. If you remember, vegetative ones are those that would have like just leaves, and they are basically responsible for the production of the food, so through photosynthesis, whereas the reproductive ones will be the one that ultimately would lead to the formation of flowers and then fruits. So they're in charge of reproductions. Now, an interesting feature is that once the axillary buds, so the axillary meristem is left behind, in some plants it stays inhibited by the apical meristem, which will be present here. So these will basically stop the growth of these cells. So this tissue will stay undifferentiated, will not be divided until the apical meristem gets removed. And that's the reason why, for example, you need to prune some plants. There are certain type of plants where you have to actually cut the main stem, that is growing upward, so to get that lateral growth of the plants. Otherwise, it would not take place. So you're removing that inhibition by the apical meristem into the axillary over the axillary meristems. So now, same growth happens in the root. Okay, so primary growth driven by apical meristem takes place in the root as well. This takes place through cell division and cell elongation, and obviously cell differentiation here. There is some slight difference in the locations of the ultimately the meristematic tissues produced by the apical meristem, and the location therefore of the final tissues produced by these apical meristems. So first of all, now the root itself, while it's expanding underground, will be subject to a lot of friction. So in this case, the apical meristem is not found right close to the surface, but is a little bit further set in, if you want, because it's protected by these bunch of epidermal cells, ultimately, which form what is called the root cap. These epidermal cells are cells that with time, through friction, would die and will be replaced, at the same time produce some lubricants too, so allowing the growing root to be not subject to excessive frictions while it's moving inside the ground. Now, when you look at the structure of a root where the apical meristem is actually active, you would see, and if you take like a longitudinal section like this one right here, by longitudinal section what we mean is you have taken a root and you have cut it following the main axis. So you're really basically dividing into a half. This is different than a cross section. So through a longitudinal section, you can actually find different region which corresponds to where the apical meristem is and where the three primary meristems produced by the apical meristem are and how they differentiate. So these three zones are referred to as the zone of cell division, the zone of elongation, and the zone of maturation. So up to here, you still have undifferentiated tissue, and here that's where the undifferentiated meristems, so three primary meristems, would actually differentiate into the final tissue. How does it look like? So in the zone of cell division, that's where you have the apical meristem. The apical meristem is very actively dividing there, right? By doing that is adding more and more cells, which then are left behind, and they will form, exactly like in the stem, the three primary meristem. So they will form the protoderm on the outside, and the procambium in this case will not be between the protoderm and the ground meristem, but will be in the center. These are eudicots. If you remember the stele with the vascular tissue, it's found in the middle of the root, not on the side. Whereas in the stem of eudicots, it will form a nice ring. So in this case, in the center, so the procambium is actually found in the center, and the ground meristem at this point is between the protoderm and the procambium. These three tissues, so the protoderm will give origin in the end to the epidermis, the ground meristem will give origin to the ground tissue here of the cortex, and the procambium will give origin to the vascular tissue, xylem and phloem, found inside the stele here. So this division here takes place in the zone of cell division. These three primary meristems are produced here, and then in the zone of elongation, now these cells here, they start elongating. They get longer and longer. By doing that, they basically cause the pushing of the root further deep into the ground. And once these cells then they reach the zone of maturation, keep in mind that they stay there, it's just that the root is growing. So it's not really that they're moving upward, it's just the root that is growing. Certain point that will become zone of maturation, I should say. And that's where they will differentiate. So you would have that the protoderm, will form the epidermis. The ground meristem will form the cortex, or the ground tissue here, and the procambium will form ultimately the xylem and the phloem here, which is found inside the stele. Together with that, you would have also the formation of a layer here called the pericycle, which is very important for the lateral growth, you see later on, and the endodermis. And these will be the ground tissue formed by the ground meristem here, corresponding to the cortex. So by doing that, now you have that the apical meristem has caused primary root growth, pushing the root deeper into the ground and forming all the tissues necessary there. Now, the pericycle here, that layer that I just mentioned here highlighted as yellow cells, is also the one that is responsible. So it's for lateral growth. Lateral growth meaning still as primary growth. So this root can actually start forming side roots growing on the other side. It's different from the root hair. The root hair are nothing else than specialized epidermal cells. In this case, what you have is that this root can form a full root moving sideways as well. How does it happen? Is because the pericycle cells, some of them are still fairly undifferentiated. And by doing that, they can actually create a new apical meristem, which then will start expanding in a different direction. So imagine this root is growing towards you. At this point, this new apical meristem originating excuse me, from the pericycle here start growing perpendicular to the root that is growing towards you. And by doing that, will cause a lateral extension of the root, which again will still be undergoing primary root growth based on the apical meristem here, and will follow the exact same process through the zone of cell division, zone of cellular elongation, zone of cell maturation, as we described earlier on. Now let's talk about the secondary growth. So we talked about the primary growth. All plants have that primary growth driven by the apical meristem. Okay. Now, some plants, the wood plants, these are eudicote plants, they would have also a secondary growth which allows them to grow sideways, meaning in terms of width, okay, increasing the girth, as we say. So now this is driven not by apical meristem, by two meristem, which together are referred to as lateral meristem, which are the cork meristem and the vascular meristem here. So there are two rings of undifferentiated tissues found in two different parts here of the stem or the roots. Both the stem and the roots of these plants would undergo secondary growth. Now, what are these lateral meristems? So one is referred to as the vascular cambium. This is actually produced by leftover procambium. We'll see that particularly in the root when we talk about later on. So it's from procambium, left behind there in the root, is generated also by the pericycle. And this, as the name says, is vascular cambium, so will give origin ultimately only to vascular tissue, so to secondary xylem and secondary phloem. Secondary growth gives origin to secondary tissue. Primary growth would give origin to primary tissue. So the xylem and the phloem produced by primary growth from the apical meristem is referred to as the primary xylem and primary phloem. The one formed by lateral growth or lateral meristems is referred to as secondary xylem and secondary phloem. Together with that, then you have the second cambia here, or second meristem here as lateral meristem, which is the cork cambium. As the name say, is the one that produced the cork, okay, and the phyloderm. The cork is what is going to be produced basically outside, and the phyloderm is what is going to be produced inside here, which ultimately will be part of the bark. We'll discuss these things as well. So now the new tissues that are produced here again are referred to as secondary tissues. Let's start talking about the vascular cambium. So now this cambium here is lateral meristem that produce vascular tissue. So how is it organized? It's organized on a ring, and this is nothing else than basically procambium left behind by the apical meristem. Now this is how a cross section of a eudicote stem will look after primary growth. If you remember, you have the vascular bundles that are nicely organized, if you see a larger picture here, forming a ring here. And now in between, so here you would have the primary xylem, these large cells here. You would have the primary phloem here. You would have some sclerenchyma here, supportive. And then in between the phloem, the primary phloem, and the primary xylem here, you have some procambium left behind. Originally, this is separate, so in each vascular bundle, the procambium is separate from the other one. But then it grows and basically start forming a full ring all around, so connecting all the vascular bundles here. And basically, by doing that, you end up having a procambium here that separates the xylem towards the pith. This is the pith, the inner part of the eudicots in these plants, and from the phloem, which will be embedded in the cortex on this part here. How does this lead ultimately to the formation or the growth, lateral growth of these plants through formation of new vascular or secondary vascular tissue, so secondary xylem and phloem? Well, what you have here is the procambium is made by undifferentiated cells, which have two big properties. They have the capacity to self-renew, meaning that they can reproduce themselves. So a cambial cells here can produce another cambial cells, and both of them will be undifferentiated. So they can produce more and more cambial cells. So it can self-renew itself. Not only that, but it can also differentiate. And so what you have is that these cells can differentiate both in xylem or phloem. So what happens here, and how do you achieve basically growth here of new xylem and new phloem? You have that these procambial cells, they first divide, the first step, and then you would have that one of the two cells stays as procambium. You don't want these both of them to differentiate, otherwise you will run out of the vascular cambium. Whereas the other cells would differentiate, the one towards the inside would differentiate into a xylem cell. And then the leftover procambial cells would duplicate again, will self-renew, and then the second cell that is produced on the outside would differentiate into phloem. This is going to happen again. So the cell that is left over, the procambial cells, will duplicate again and now produce another xylem cells towards the inside. Duplicate again, produce another phloem cells. By doing that, basically it's producing more and more xylem towards the inside and more phloem towards the outside. And these will be referred to as secondary xylem and secondary phloem. And this is how we will look at this point, this plant. So this is the plant, how we will look after primary growth. So you would have a primary phloem, primary xylem here. And now you have that the primary phloem has been pushed towards the outside, and you have secondary phloem appearing in contact with the vascular cambium. And on the inside, instead, you have that the primary xylem has been pushed toward the middle, or actually stays where it is in the end. It doesn't really get pushed. But then you have a secondary xylem that's been produced in contact with the vascular cambium. By doing that, basically you have achieved, since you're adding more cells, you're achieving growth of the plants in this direction. Okay. Obviously, this is gonna extend the plant in this direction, but it's gonna put strain to the surface of the plant. This plant could not sustain growth and growth and growth in this side without breaking the epidermis ultimately. So first of all, this is a picture where you can see actually the two cambia. So this is where the vascular cambia will be. That's actually a 3-year-old tilia. This is a wood plant, produce actually a tree. And you can see this is the vascular cambium, this straight line here. All you see towards the inside is xylem. This would have been, so this is 3 years old, so this would have been the primary xylem, the one formed by primary growth, and all of these are secondary xylem. So this was produced on the 2nd year, this on the 3rd year. And on the other side of the vascular cambium, here you have, now so this will be the vascular cambium by the way, you have instead the phloem that's being produced, so that with the primary phloem being towards the outside and the secondary phloem here towards the inside. Together with that, now you have a second type of cambium here, referred to as the core cambium, and that's the one that is responsible for basically keep replacing or expanding the surface outside, which otherwise will break with this constant growth of the tree outward. So the core cambium here, so that's the one we're talking about right now, the one on the outside. This has the responsibility to to replace ultimately the outside, the surface of the plant. So the epidermis and all the ground tissue that's present in there. This is originated, so here you would have the apical meristem is originated from some ground meristem that is left behind, okay, only again in wood plants. And this, it will form ultimately, so it's found in the cortex. So the ground meristem here would have produced the pith and the cortex, and some of these tissues found in the cortex. That's the one that forms the core cambium. Now, this core cambium is also referred to as phellogen. Okay, it will be, that's how it looks here. That's the phellogen, and now produce two type of tissues similar to the vascular cambium. Produce cells both towards the outside and the inside of the plant. So toward the outside, produce what we call the cork or phellem. So this is cells that ultimately are gonna be very rich. They have a very thick secondary wall impregnated with lignin and suberin covered with wax. So these are cells that ultimately are gonna form a very strong, dense structure. That's exactly what you see on a tree, on a wood tree, right? Try to punch it, try to break it, you can scrape it out, but you can't really break it very easily. You need a saw to cut through this. Okay, so that's referred to as the cork. Okay, and towards the inside instead produce these secondary cortex, which is ground tissue, which is called phelloderm here. By doing that, it's like the dermis that you have under your skin. Imagine. So you have the core cambium, and this again contributes to the lateral growth, and most importantly contributes to expanding the surface so that it can keep up with the growing, the lateral growth of this stem right here. Now, in terms of words, so when you put together the cork, the cambium, and the secondary cortex, or the phelloderm here, so basically the core cambium and whatever it produces, what you're talking about is referred to as the periderm here. Okay, similar to basically imagine your skin. You have your skin there, skin cells on the top, you have dermis and basically connective tissue. And then when you add to the periderm also the xylem that's being produced, basically the secondary, phloem, that's being produced by the vascular cambium. So from here all the way outside, so you would have the periderm, and you add the phloem here that's being produced, the vascular cambium here, you're talking about the bark. Okay, these are the words that are normally used. Now you can see how ultimately, if you're working with, or you have some wood tree in your backyard, what you should do should never remove the full bark, because if you're removing the full bark, you're actually removing also the phloem, which is that tissue that has to move the nutrients throughout the tree, right? So you're gonna kill the tree. So all you can really remove is just the cork. So basically up to the cambia here, although this would cause, you could only basically remove only the cork, meaning the phellem, not the core cambium. Now, the inside, whatever it's inside of the vascular cambium, is referred to as wood. That's what you like to buy at Home Depot. All right, and now there are two types of wood. There is what is called the earthwood and the sapwood, and you can see it with the coloring. So now, what you see here is actually this area enlarged. Whatever you see dark on the outside would just be the bark. So your vascular cambium will be at this level. All of these is nothing and the xylem. Primary xylem towards here, and then all secondary xylem. The secondary xylem can be dark, so part of the sapwood or the earthwood. What happens there is that while the wood is produced, the plant is growing this way. Now you have that the xylem, progressively, the one towards the center, tends to be saturated or start dying and be saturated with antifungal and antibacterial molecules. So you don't want insect to be able to go through this. Now, by adding also all of these antifungal, antibacterial molecules, this structure becomes very hard. And so it's going to help at this point the growing tree. Keep in mind, this tree is growing taller and taller and taller, right? So it could easily break. So now these earthwood here produce this very strong structure made by dead cells, which helps giving rigidity ultimately to the tree. And then on the outside you have the sapwood. This is actually the active xylem. This is still moving water. Okay, so these are all live cells which are present in there. This is what you see in these beautiful pieces of furniture, for example. This dark one, this part here will be the earthwood. What you see outside here is the sapwood. So as you can see, the dimension can change depending on the tree you're talking about. Again, summarizing all the words that we use here, we're using a lot of words. So we talked about lateral growth. So we talked about two type of cambium or lateral meristem. We talked about the vascular cambium here. And then you would have here, you would have the core cambium here. Now, the cork produces the cork on the outside. So the core cambium produces the cork on the outside and the secondary cortex here towards the inside, which is referred to as the phyloderm here. When you put all of this together, you're talking about the cork there. Now, when you add so now you have the vascular cambium, so that is producing the vascular tissue, so it's producing xylem towards inside. This is what is referred to as the wood. You would have the primary xylem in the middle, and then all secondary xylem forming here. The one that basically has died and is being filled up with antifungal, antibacterial stuff, and so on, is referred to as earthwood. And then the one is still active, lighter in color, is referred to as sapwood. And then on the outside of the vascular cambium, you would have the phloem, secondary phloem and primary phloem. When you include the phloem to ultimately the periderm here, so the core cambium and secondary cortex, you get what we call the bark. Now, as you're very familiar with, you know that if you actually do a cross section of a tree, you start seeing a series of circles. These circles, they do correspond to basically the different cycle of lateral growth of the tree. Now, lateral growth can take place throughout the whole year. Okay, and you have spring growth and fall growth. Now, these rings, so a full cycle, a full year would include both a light ring like this one and a dark ring. This is the combination. The light one is what you refer to as the spring growth. Is that growth that takes place in the spring and also in the summer when there is more water. For this reason, the lighter areas tend to be much larger in general, usually in general, than the darker ones. Okay. Now, not only that, but actually the width of this area reflects also the amount of water is present there. These, again, are made by usually large xylem cells, vessels, and the width of the vessels there can actually be larger or smaller depending on the amount of water is present during that spring growth. And then this is the fall growth. Okay, so there is less water available, less growth. These cells tend to be smaller in width. These vessels are narrower and therefore more dense, giving this darker appearance there. And so by counting, obviously, as you know, all of these rings, you can actually figure out how many years, so a combination of a light and a dark one corresponding to 1 year, you can figure out how many years have passed and get also information on how the season was. Was it a very wet season on that specific year or not? Now, let's finish up talking about the secondary growth in root. So we talked about the secondary growth in the stem of wood plants, but what happens in the root of eudicots or wood plants, part of eudicots? So this growth, lateral growth, takes place in roots as well. Now, it takes place overall in a similar way as in the stem, however with some minor differences. First of all, the vascular cambium in this case, so the lateral meristem here consists as well of vascular cambium and of cork cambium. The vascular cambium in this case, so this is how a still, so where basically all the vascular cylinder, so the vascular tissues is organized within a eudicot. So you would have the cortex here, and then you have the still here, which then where you can identify the endodermis, the pericycle here, and then you have the xylem, which are large vessels here, and the phloem here. Now, in between the phloem and the xylem here, there is some procambium that is left behind. Okay, similar to what was found also in the stem. However, this procambium is not in continuity. And what you have here is that the pericycle cell here, this single layer of cells here, now in this area here still has meristematic properties, and so it actually goes in continuity with the procambium that is left behind here, forming basically a layer of vascular cambium all around the xylem. Again, if you remember, it's not going to be a circular structure here, but so it's right here. It will form, again, it will still isolate the xylem towards the inside from the phloem towards the outside. And that is what acts in the root as the vascular cambium. So similar to exactly how it was happening in the stem, now you have that the vascular cambium now will produce xylem towards the inside and phloem towards the outside. By doing that, is basically forming a ring ultimately, okay, where you have the primary xylem here and all secondary xylem here. So these will be the vascular cambium now at this point after a few seasons. And then you have here the secondary xylem, phloem, sorry, that gets produced towards the outside by the vascular phloem, similar to what's happening on the stem at this point. The only difference there will be the originary structure of the vascular cambium, which is not forming a nice circle as before. But then ultimately turns into a circle here, and now the growth, side growth, is basically gonna crash the cortex. So this tissue here will get pushed more and more towards the outside and crash and basically slough away. Now again, in the root as well as in the stem, this is going to put straight into the surface, and so the surface has to be replaced too. And so there is a core cambium here as well, which is indeed produced by the pericycle. So the pericycle, again, the same pericycle that he is producing also, this vascular cambium here, is also producing towards the outside. It's not the yellow tissue, it's the endoderm, but it's going to produce the core cambium here towards the outside. And this core cambium works exactly like in the stem. So this phellogen produces the phellum, the cork, towards the outside, and the phelloderm towards the inside. And by doing that, basically keeps replacing the surface so that the plant can be protected from the external agents. So summarizing here, what did we talk in the first and second part of this lecture? Well, first of all, we went through the structure of the plant cells, and particularly focused on the unique features of the plant cells compared to the animal cells. Particularly, we talked about the structure of the primary and the secondary cell walls. And we say also, later on, we talk also about where you find the secondary, this lignin-enriched type of cell wall. Then we talked about the tissues and the organs. So we talked about the three main tissue types that you find in plants: the ground tissue, the vascular tissue, and the thermal tissue, and all the subtypes of ground tissue and vascular tissue that you find in there. And then we talked about how they're organized, for example, in organs such as the leaf. Now we talked about the anatomy of the plant. We defined what the stem, or the shoot, sorry, and the root systems are. So we talked about then about the stems and the roots, and we focus on the difference between these two big groups of angiosperms, which are the monocots and the eudicots. What is the difference in anatomy, in leaf appearance, in this distribution, for example, also of the vascular bundles within the stem and the roots? And then finally, in this second part, we talked about the growth of plants. We talked about how plants can undergo both primary and some of them secondary growth as well. And we talked about the meristems that are responsible for this growth. So we talked about the apical meristem and the axillary meristems, which is generated by, is basically apical meristem. We talked about the intercalary meristem, and then we talked about the lateral meristem. So then we talked about primary growth, how it's sustained in all plants by apical meristems, and in some monocots by the intercalary meristem, and then how this apical meristem produce all these three primary meristem, which are the procambium, the protoderm, and the ground meristem, which then will form all the different tissues in both the the um, the roots and the shoots, or the stems, sorry. And then we talked about, finally we finish up talking about secondary growth, which takes place only in wood plants, and we saw how it takes place both in the stem and in the roots. And we talked about the lateral meristems that are responsible for this growth. So we talked about the vascular and the core cambia and how they work to produce, to enlarge ultimately the stems and the roots in the plants that undergo secondary growth.